This chapter 141 concerns the neuropathology of movement disorders. These are classified into four major groups according to their clinical phenomenology. Hypokinetic rigid forms like Parkinson's, hyperkinetic forms like chorea, atactic movement disorders like cerebellar ataxias, and motor neuron disorders. However, this specific chapter only concerns the two first forms, hypokinetic and hyperkinetic forms. Before this, I would briefly discuss the functional anatomy of basal ganglia, because most of akinetic and hyperkinetic forms have their origin in the dysfunction of the basal ganglia, a highly interconnected set of subcortical nuclei, input, output, and intrinsic nuclei. Basal ganglia are a network of sets of multiple parallel loops where cortical and subcortical projections interact in tandem with the cortex through complex circuits of the brain. The interconnection of these nuclei are shown schematically in the following slide. Here you have the functional anatomy of the basal ganglia, which speaks for itself. Activity of the basal ganglia output structures is controlled by two opposing striatal motor loops, direct inhibitory pathway and indirect disinhibiting pathway, projecting to the basal ganglia. Both are structurally and functionally regulating movement inhibition and execution. Two hyperdirect pathways include excitatory communications from the cortex to the subcortical and to the internal pallidum, resulting in inhibition of movements. The various movement disorders result from dysfunction of cortical, basal ganglia, thalamocortical circuits due to disruption of downstream network activities in cortex, thalamus, and brain stem. Here you have the functional anatomy of the basal ganglia with a schematic diagram of the basal ganglia thalamocortical circuitry under normal conditions and in Parkinson's disease, with the different chemical substances which are working in this functional cooperation. Now again, the classification of movement disorders based on pathophysiologic mechanisms are Parkinsonian syndromes, choreoabolism, and dystonia. But based on recent genetic and molecular biological data, movement disorders are classified in the following form: A, alpha-synucleinopathies, tauopathies, TDP-43 proteinopathies, polyglutamine repeat CAG disorders like Huntington's, and a number of other hereditary disorders. Here is a model of protein misfolding and fibrillation leading to the deposition of aggregated proteins in cells and extracellular spaces with forming of intracytoplasmatic inclusions like neurofibrillary tangles or Lewy bodies. Here you have another classification of movement disorders with a cascade of neurotoxic effects of protein oligomers leading to neuronal dysfunction. You have microglia, astroglia proliferation, you have A-beta or synuclein oligomers, you have mitochondrial dysfunction, which all act together with neuroinflammation, synapse degeneration, axon transport dysfunction, apoptosis, exotoxicity, and they are finally leading to a neuronal dysfunction and neurodegeneration. Here you have the neurodegenerative disorders with protein deposits, the abnormal deposits, extra, intracellular, and internuclear, with the specific types. Alpha-synuclein, tau, we have 3-repeat and 4-repeat tau pathologies, TTP43 and others which are rather rare disorders. Now, alpha-synuclein is a schematic representation of the structure, protein domains, and phosphorylation sites of human synuclein, which is composed of three distinct regions, as you see here: the alpha helical N terminus, the NAC, and the addicted C terminus. The pathologic deposition of alpha-synuclein is formed to be the so-called Lewy bodies or Lewy bodies. In B, you have cortical Lewy bodies. In C, you have an ultrastructural picture with a center and with a periphery. In D, you have Lewy neurites, and in E, you have a number of various cortical Lewy bodies with a specific immunohistochemical stain. Lewy body diseases are staged according to various forms. The most frequently used is the Braak stage 1 to 6, beginning in the medulla and the anterior olfactory nucleus, going upwards to the medulla oblongata, midbrain, the mesencephalon, and finally the neocortex. This is a schematic representation of the Braak stage 1 and 2, initial in the medulla and in the olfactory bulb, then stages 3 and 4 going to the midbrain, which is associated with sleep and motor disturbances, and as Braak stages 5 and 6, finally involving the cortex, and they are associated with emotional and cognitive disturbances. There are other classification forms, for instance, by Beach and co-workers, which are shown here. It begins in the olfactory bulb, goes either to the brainstem or to the limbic areas, or to both, and finally to the neocortical areas. Very recently, another classification has been proposed by a group with Adams and co-workers, which is in press in the acta Neuropath. Here you have the Newcastle McKee's revised criteria of the Lewy-related pathology. You have the category of Lewy-related pathology, olfactory only, amygdala predominant, brainstem predominant, limbic and neocortical, and here you have the classification criteria, which are now have been published by Adams and co-workers in acta Neuropath. Then we come to the progress and distribution pattern of pathology related to Parkinson's disease. We have here again stages 1 to 6, which are shown here with different colors. Here we have the schematic diagram of the basal ganglia thalamocortical circuitry under normal conditions to the left in hypokinetic and hyperkinetic movement disorders. The next is Parkinson's disease without treatment of DOPA, and on the right side you have the scheme of levodopa-induced dyskinesia. The next model shows the several mechanisms underlying resting tremor in Parkinson's disease. The major pathways of the pathogenesis of Parkinson's disease are shown here. You have endogenous factors like alpha-synuclein, genetic factors, and exogenous factors, which all together finally cause protein aggregation, energy dysfunction, the production of Lewy bodies, and finally ending in neurodegeneration. Now, causes of Parkinsonism in general. Common causes of neurodegenerative forms are idiopathic Parkinson's disease, multiple system atrophy, dementia with Lewy bodies, progressive supranuclear palsy, and genetic diseases. And then we have a large number of uncommon neurodegenerative causes of Parkinsonism, for instance, vascular, corticobasal degeneration, and so on. Here we have dementia with Lewy bodies in comparison to Parkinson's disease dementia. It has been discussed for a long time whether these are two different diseases or two subtypes of disorders with production of Lewy bodies. We have both clinical and neuropathological similarities and dissimilarities. The similarities are that both produce extrapyramidal symptoms and dementia, but their differentiation is according to the so-called 1 year proposal. That means that Parkinson's disease dementia first begins with extrapyramidal symptoms, and after more than 1 year, development of cognitive dysfunctions, dementia, whereas in dementia with Lewy bodies, usually cognitive disorders and psychiatric side effects are in the beginning of the disorder, and after 12 to 24 months, there's development of cognitive dysfunction and dementia. Here you see the continuum of Lewy body disorders, which are beginning with very few deposition of Lewy bodies. Then we have Parkinson's disease non-demented, Parkinson's disease dementia, dementia with Lewy bodies, dementia with Lewy bodies combined with severe Alzheimer pathology, and finally Alzheimer's disease. We know that most cases of dementia with Lewy bodies are associated with considerable Alzheimer's disease. These are preliminary neuropathological criteria for differentiation of dementia with Lewy bodies and Parkinson's disease dementia. Here you see that both are characterized by combination of progressed Lewy body pathology and variable changes of Alzheimer's pathology. Alpha beta load tau phases are very similar, whereas tau load is much higher in dementia with Lewy bodies than in Parkinson's disease dementia. Further minor differences are that the cortical Lewy body load is higher in the temporal and parietal cortex in dementia with Lewy bodies, but in particular the hippocampal subarea CA23 is more severely involved, and there are slight differences in the neuronal cell loss of substantia nigra. Next is multiple system atrophy. Here you see mild diffuse atrophy and a discoloration, bluish discoloration of the striatum. Histological characteristics are the so-called glial cytoplasmic inclusions, which are composed of alpha-synuclein and many other chemical substances. Here you see it in the in the globus pallidus or in the white matter. This is the schematic distribution of various combination types of multiple system atrophy, which previously has been differentiated in so-called striatonigral degeneration and oligopontocerebellar atrophy. And here you have the variation of different severities of pathological lesions in the different forms. The putative pathogenetic pathways of multi-system atrophy are, in general, it is a sporadic disorder. Very probably we have begin with either neuronal dysfunction or oligodendroglial dysfunction, which is combined with pathologic aggregation of alpha-synuclein, dysfunction of oligodendroglia, together with neuroinflammation, oxidative distress, causing a degeneration of oligodendroglia and neurons. It has been discussed whether MSA is a primary oligodendrogliopathy, that means a primary dysfunction of oligodendrocytes, or it is a combined oligodendroglial neural dysfunction. However, very recent animal studies have been shown that very probably a primary oligodendroglial dysfunction is essential for the pathogenesis of this disorder. Then we come to so-called tauopathies. Here you see the various tau isoforms. We have 6 different isoforms, and here you have their correlation of the various isoforms to various tauopathies. On one hand, we have Alzheimer's disease. Then we have this is a combination of 3 to 4 repeat tau. Then we have so-called 4 repeat tau, PSP, CPD. Then we have 3 and 4 repeat taus, and they are very different in their classification. Here you have the tauopathies, progressive supranuclear palsy, a 4 repeat tauopathy, the same cortico-basal degeneration. Then the amyotrophic lateral sclerosis, Parkinson dementia, complex of Guam, which has a 3 and 4 triplet. The same is with post-encephalitis Parkinsonism. Then we have a number of other tauopathies, pallido-pontonegral degeneration, Pick's disease, which is mainly a 3 repeat tauopathy. Then we have other forms, amyotrophic lateral sclerosis, Perry syndrome, and so on. And frontotemporal lobe degeneration with MAPT maturation. This is the tau-related gene. Next are so-called TTP43 proteinopathies, which are mainly different types of frontotemporal lobe degeneration and also amyotrophic lateral sclerosis. Here, an example of progressive supranuclear palsy with hypomimia and with hypermodulation of the eyes, mainly dysfunction of upper and down gaze. And here you have the localization of the pathological depositions in progressive supranuclear pathology. Here we have a schematic diagram of the basal ganglia thalamocortical circuitry under normal conditions again. Parkinson's disease of levodopa-induced dyskinesia. Then we have multiple system atrophy, which is slightly different from Parkinson's disease. Then we have PSP, we have Huntington's disease, we have dyskinesia, and we have other types of movement disorder. Here you have the normal cortico-basal circuitry, supplementary or primary motor cortex, substantia nigra, putamen, globus pallidus, thalamus, and the others, and with the different chemical substances which are working here. Here is a schematic pictures of the pathology of hippocampus in fixed disease, which differs from that, for instance, in Alzheimer's disease. Here you have frontotemporal lobe degeneration with tau, which is classified into several subtypes, like PIC disease, PSD, cortico-basal degeneration, global glial tauopathy, adrenophilic brain disease, and frontotemporal lobe degeneration with MRPT mutation. Here we have secondary causes of Parkinsonism, symptomatic forms, which we have already shown before. Here is again the schematic diagram of basal ganglia thalamocortical circuitry under normal conditions, and with Huntington's disease, with dyskinesia, and with hypokinetic rigid disorders, Parkinson and myoclonus. Here we have hyperkinetic disorders. We have two major groups, the polyglutamine repeat CAG disorders, for instance, Huntington's disease, Mercado-Joseph's disease, chorea acanthocytosis, and so on. And then we have other hereditary degenerative disorders, for instance, hereditary degeneration, Hallervorden-Spatz disease, and so on. Causes of chorea, we can differentiate between hereditary and sporadic forms, hereditary, Huntington's disease, neuroacanthocytosis, and so on, ataxia telangiectasia. The major forms of sporadic are Sydenham's chorea and chorea gravidarum. Here are the direct and indirect pathways in chorea, motor, striatum, thalamus, globus pallidus, and all together are forming the pathways, facilitatory and inhibitory ones, which are affected in choreatic syndromes. This is the dysfunction in Huntington's disease. We have the communication between cortex, thalamus, striatum, and the basal ganglia. Next group, which are rather rare disorders, are pallidal degeneration. Here is a large list of various types of these rare, mainly autosomatic recessive or sporadic disorders. Here you have neurodegeneration with brain iron accumulation type 1, previously called Hallervorden-Spatz disease, with the discoloration of the globus pallidus, as seen with the arrows. Next is neurochemical pathology of Parkinson, which is an interplay between cerebral cortex, striatum, and thalamic cortical. Next, pathophysiology of dystonia. This is characterized by sustained contractions of agonistic and antagonistic muscles. We have no consistent morphologic abnormalities for primary dystonia. There are several structures involved, putamen, thalamus, premotor cortex, but also spinal cord and peripheral nerves. The digestive pathophysiology is a loss of D2 receptors in the putamen, with changes in neural activity in the striatal circuits, and a decrease of lymph discharge rate of globus pallidus, and disrupted cortical functions probably a combination of all these disorders are the basic pathophysiology of dystonia. Here we have phenomenology of rest tremor with the major parts rest tremor, non-voluntary activity, which is typical for Parkinson's disease. Then we have action tremor, postural kinetic tremor with various subtypes. Here is a syndromatic classification of tremor. Physiologic tremors, which are rather rapid, enhanced physiologic in essential tremor, which is very often of unknown cause, or it may be a genetic form. Then we have dystonic tremor forms. Then we have the Parkinsonian tremor, postural and kinetic, cerebellar tremor, Holmes tremor, palatal tremor, which in fact is myoclonus due to symptomatic brainstem cerebellar lesions. We have drug-induced or toxic tremor and others, and finally physiogenic tremor. In conclusion, we can say that despite considerable clinical and pathologic overlapping, most types of movement disorders show characteristic pathological pictures. The deposition of pathologic fibrillary proteins in the distributing pattern of CNS lesions may or may not be typical cytoskeletal signposts pointing to the correct diagnosis and to the pathophysiology. Because in vivo markers for the most of these disorders, except for those with known molecular genetic backgrounds, are lacking, the diagnosis usually depends on clinical morphologic features and, in final, by neuropathology. Specific identifications and correct diagnosis of some of these disorders may be difficult because many of them share clinical and morphologic phenotypes with other neurodegenerative diseases, for instance, Parkinson's and multiple system degeneration, or Parkinson's dementia, dementia with Lewy bodies, and so on. Consensus data on clinical and neuropathological criteria, together with molecular genetic and biochemical data, will aid in correctly classifying and diagnosing neurodegenerative movement disorders, and hopefully will provide further insight into their pathophysiology and pathogenesis as a basis for future preventive or therapeutic strategies. Thank you very much for your attention.